A very good morning, all of you. This is Dr. G. Manigandan, working as Assistant Professor and Head, PG and Research Department of Biotechnology, JJ College of Arts and Science, Autonomous, Pudukkottai. Now, I am going to discuss you about history of immunology. That is nothing but the milestones occurred in the field of immunology, or else the contributions of various scientists in that discipline. This slide shows you what you mean by immune and immunity. The adjective immune comes from the Latin word immunis, which means exempt from public service. If you are protected or exempt from various disease, injury, work insults, then you are immune. The next one is immunity, a condition of being able to resist a particular disease, especially through preventing the development of pathogenic microorganism. Existing belief. Disease and pestilence, that is nothing but a fatal epidemic disease, where punishment rendering as a result of bad deeds or evil thoughts. Old Testament is filled with pestilence. Old Testament, in the sense, it is nothing but the first part of the Bible. It is filled with a pestilence, a fatal epidemic disease. So, a disease was regarded as a punishment given by the God. And this slide shows you the observation made by Thucydides, a Greek historian, recorded about the disease plague in Athens. Plague, a bacterial disease which is caused by a bacterium Yersinia pestis. About his observation, those who contracted the disease and recovered were the ones who could tend to the sick and dying without worrying about catching it again. That means those who have suffered and affected from the disease, and the disease will not be coming again in the future. He is talking about the attenuation, a form of immunity. Necessity is the mother of invention, a great proverb saying by Plato. It is all about the smallpox vaccine. The smallpox was probably the greatest single incentive towards the precipitation of modern Western science and to the onset of the field of immunology. Historically, the smallpox has been a worldwide terror killing millions of people. This slide shows you the symptoms of smallpox. The first symptoms are a high fever, chills, backaches, and headaches. Day 4, it will be producing rash on face, chest. In day 7, it will be producing blister-like pustules. And in day 9, pustulates split open, dry up, and form scabs which fall off and leave scars. If the virus attacks the eyes, lungs, throat, liver, it can lead to death. And this slide shows you the symptoms of smallpox, the pustules. And now coming with a variolation. In the year 1400 in the Middle East, the practice of variolation was common. What do you mean by variolation? Applying powdered smallpox crust and inserting them with a pin or poking device into the skin. So that is the variolation. Applying powdered smallpox crust, the sense this is the outer layer, and inserting them with a pin or poking. The meaning of poking is pushing or prodding, like that. So them with a pin or poking device into the skin, that is the insertion. So a process of controlled infection, also called as inoculation. In 1721, Lady Mary Wotlow Montagu, the wife of British ambassador in Istanbul, helped to popularize inoculation. She had her son, 6 years old, and 3 years old daughter, inoculated by the surgeon of embassy for fear of the deadly smallpox epidemic. And this is the history of the discovery of the smallpox vaccine. As we all know, the proverb, a thorn can only be removed with another thorn. A diamond can cut another diamond. As like that, a virus disease can be prevented by another virus. A virus can be prevented by another virus. So that is a concept for the smallpox disease. So the use of cowpox virus against the smallpox. A first step to a safer procedure was to substitute the material which is derived from the lesion of a cowpox for the inoculation. The cowpox, it is a benign disease which is caused by a virus which is closely related to the smallpox virus. So it is closely related virus, cowpox and smallpox. In 1774, Benjamin Jesty, a farmer who inoculated his wife with the vaccinia virus. And this is the first record of anyone using vaccinia virus to protect against smallpox. And now the discovery of Edward Jenner in 1796. He was tested by whether the belief that cowpox sufferers were actually immune to smallpox was true. His first experiment, a boy inoculated with a pus from a cowpox sore, and the result, the boy caught the cowpox. After 3 months, the boy was inoculated with a pus from a smallpox sore, and the result is the boy did not catch the smallpox. And now Jenner called this new method of inoculation, what do you mean by it is vaccination from a cow, vacca, that is vaccinia virus that cause cowpox, as a way of distinguishing it from the process of inoculation. So Edward Jenner performing the first vaccination against smallpox in 1796. So one of a great milestone. So in the field of immunology, he was the first person discovered the vaccine. So he was the pioneer in the field of immunology, and he was also said to be a father of immunology. And now the contributions made by the Louis Pasteur, a great biochemist and a microbiologist. He developed the germ theory of disease. What do you mean by germ theory of disease? A disease caused by a microorganism. A germ theory of disease proposed that most diseases are caused by microorganisms transmitted from an infected individual to a non-infected one. So the Louis Pasteur became the first experimental immunologist. He developed so many attenuated vaccines against anthrax, cholera, and rabies. So anthrax and cholera are the bacterial disease, and the rabies, it is the viral disease. So like that, the modern vaccines against disease such as measles, polio, chickenpox are based on this principle. A great discovery by the Louis Pasteur, and this slide shows you the discovery of the cholera vaccine. He take the aged bacterial culture and he injects into normal healthy chicken. So after 1 week, no disease will be developed in the normal chicken. The chicken remains normal. And then in the next stage, he take the fresh bacterial culture. If it is fresh in the sense, the organism, it is uh, potential, that is, it is in the active log phase. So he injects that the fresh bacterial culture to the immune chicken. That means the chicken that is previously injected with the aged bacterial culture. Now after a week, the immune chicken, it remains live. So live chicken. Why? Because even though the pasteur inject a fresh bacterial culture, see the injected the host, it is immune chicken. The chicken that is previously injected with the aged bacterial culture, and that aged bacterial culture, it gives a protection and it gives an attenuation, so that whenever the pasteur inject the fresh bacterial culture to the chicken remains live. And he injects the fresh culture to the normal chicken. The chicken dies because of the potentiality or the virulence of the fresh bacterial culture. So this experiment clearly demonstrates that the process of attenuation, a very interesting discovery made by Louis Pasteur. So, most heated debates in the field of biomedical sciences. Uh, One is the cellular theory, and another one is the humoral theory. A cellular theory of immunity is due to the existence of phagocytic cells within our body. What do you mean by the phagocytic cells? Nothing but the macrophages. So, these macrophages will engulf the foreign particles and destruct the foreign materials or the foreign antigens. That is nothing but the phagocytosis. And the involved cell, what do you call it, is the phagocytic cells. And the next one is the humoral theory of immunity claimed that only soluble substance in the blood and other fluids could destroy the invading organisms. And now this slide shows you the cellularist. In 1888, Emil Mechnikov, the main exponent of cellular theory of immunity, a great discovery made by Emil Mechnikov, and he was the first scientist discovered the phagocytosis process. Phagocytosis is nothing but the engulfing of a foreign particle by a macrophage. A great discovery in the field of immunology, so that he was awarded a Nobel Prize also for that discovery. He demonstrated that certain blood cells could ingest the foreign materials. And hematologist Paul Ehrlich, a pioneer in the field of humoral theory of immunity, and he proposed the most plausible humoral theory of antibody formation, the side chain theory. The theory states that the antigen, the specific antigen, reacts with specific B cells, so that antibody will be producing. So that is the side chain theory. So followed by so many scientists, and they have contributed much in the field of immunology. So in 1888, Ross and Erzin both discovered the diphtheria toxin, and in 1819, Emil von Behring and Kitasato demonstrated the presence of anti-toxins in the blood of individuals recovering from diphtheria. In 1991, von Behring used serum to treat disease and discovered immunity against diphtheria and tetanus. In 1894, Jules Bordet, he discovered a complement. It's nothing but the heat labile protein, which is involved in the antigen antibody interaction. And in 1897, Robert Koch discovered the precipitants. followed by so many contributions. So, other events soon served to catapult immunology from the microbiology labs. In 1901, von Behring was given a Nobel Prize for his work on what we call serotherapy today. In 1903, Maurice Arthus he described the localized allergic reactions. So that is nothing but the hypersensitivity. He called that reaction as Arthus reaction or Arthus response. In 1905, von Percute he shocked the world when he provided the evidence that immune responses can be deleterious. He was studying serum sickness, a form of allergy. In 1958, scientists Sir McFarlane Burnett and Niels Jern proposed the clonal selection theory, and in 1960s, Porter and Edelman enzymatically digested the antibodies, and we learn about the chemical structure of the antibodies. In 1978, Susumu Tonigawa discovered the genetic principles underlying the generation of antibodies with different specificities. In 1987, he was awarded Nobel Prize for this contribution. And now in 2000s, in 2005, the development of human papilloma virus vaccine discovered by Ian Fraser, and in 2016, a third class of immune checkpoint inhibitor, that is anti-PD-L1, it is approved for the treatment of bladder cancer. And in 2017, the Indian government approves Apicidan, the first potentially curative dendritic cell vaccine for the treatment of prostate, ovarian, colon, and lung cancer, and it is developed by the Indian biotechnology company APAC Biotech. A great contribution, and these are all the milestones occurred in the field of immunology. So, the major contributions by Louis Pasteur, Elie Mechnikov, and some of the scientists. And thank you, all of you, for the patient listening.